In the previous class, we have seen what is the cause of natural selection. There are variations between organisms in a population. Remember that the basic reason of these variations are mutations. These variations may provide higher reproductive success to one organism compared to another. If the organism manages to produce more offsprings, the variations are inherited or transmitted to the next generation. And subsequently, it leads to the natural selection of that trait. What is the main component of evolution? It is genetic variation. No other variations can be transmitted across generations. Basically, evolution is the genetic change taking place in a group of organisms. Many changes may take place in an organism. For example, the skin color of a person may change when he is subjected to different levels of sunlight. But it doesn't make a change in his genome and hence it is not transmissible. Therefore, changes will not contribute to evolution. Next important point is evolution takes place only in populations of individuals. Changes happens to individuals, but evolution takes place only on groups. No single individual evolved upon its lifetime, but a population of individuals across generations get evolved. That is why evolution is more visible on a larger time scale. Next, we shall discuss more about how variations leads to evolution. Evolution can be described as a two-step process. First, genetic variation arises in a population. It is because mutation and recombination events creates new genes as well as new combinations of genes. In sexually reproducing organisms, meiotic cell division plays a major role in adding genetic variation. Independent assortment of chromosomes and crossing over events create the new combinations of genes. In the next process, the frequency of these genetic variants increase or decrease in a population. There are several evolutionary forces acting on this frequency change. They are mutation, migration, genetic drift and natural selection. We will discuss the molecular mechanisms of mutations and recombination in a later session. Here, we will limit our discussion to the evolutionary forces acting on the decrease and increase of genetic variants. Before going into details, we will have a short introduction on the measure of genetic variation. Consider a gene encoding for a flower color. There will be a precursor material or a raw material to produce color pigment. The gene produces an enzyme which will convert the precursor to an active product, the pigment. Consider the case where a mutation happens to the gene. A nucleotide may be deleted or may be replaced by another, and the new gene type is not able to produce the enzyme. In that case, the plant will not produce colorful flowers. We call the two variants of the same gene as alleles, or alleles are different forms of a gene. Here we have two alleles for the flower color. In nature, a trait may have more than two alleles. A diploid individual will have two alleles, but the whole population may have more than two alleles. The number of each allele in a population is called allele frequency. You may denote two alleles as capital letter and small letter, and if there are more than two alleles, you can denote it as A1, A2, A3, etc. In this picture, you can see two genes A and B. Their corresponding alleles are denoted as small letter A and small letter B. In the figure C, you can see how new combination of alleles have formed due to the crossing over event. Have a look at the figure D. Please note that the first and last daughter cells has the same allelic combination as that of the parents. If you look at the second and third daughter cells, you will see a different combination of alleles. Such offsprings who have a genetic combination different from any of their parental combination is called recombinant progeny or cells. By measuring the allele frequency, we can estimate the genetic variation in a population. The more the alleles are present in a population, more the variation. Next, let us understand how does the genetic variation increases the fitness of a population. Take an example of a group of bacteria in a Petri plate. There are two populations of bacteria. The first population contains bacteria that are susceptible to antibiotic A but resistant to all other antibiotics. The second population contains three types of bacteria that are susceptible to three different antibiotics and resistant to others. If antibiotic C is applied to the two populations, some individuals in the second population will die, but all the individuals in the first population will survive. When antibiotic B is applied, again some individuals in the second population will die, but all the individuals in the first population will survive. Now, consider the case when antibiotic A is applied. Some individuals in the second population will die, but none in the first population will survive. The individuals may have differential benefit in each case, but for the bacteria as a population, it is better to have more genetic variance. The survival or the fitness of the population as a whole is dependent on the genetic diversity. This is a simple example, and there may be case-to-case differences in different scenarios. But in general, for a population, they are more fit if they have more variance in them. Now, we shall discuss what are the major evolutionary forces that act on allele frequencies. The first one is mutation. Mutation is the root cause for all variation, and it also changes the allele frequencies in a population. Consider a mutation happens to allele A1. It changes to A2. This mutation we can call as forward mutation. If a mutation happens on the allele A2, which will convert it back to A1, we call that mutation as reverse mutation. Next, see what is the effect of forward and reverse mutations. In this population of individuals, there are only A1 alleles. What is the frequency of A1 allele? It is just 1. Usually, we denote allele frequencies by P and Q. Here, 100% of the individuals have A1 alleles, so P is equal to 1. A mutation happens in some individuals, thereby converting A1 allele to A2 allele. Now, there are 6 individuals in the population with A2 allele. So, there is a change in the frequency of both alleles. Now, P equal to 0.75 and Q equal to 0.25. Again, when time goes on, some reverse mutation happens and some individuals get back the function of A1 allele. Now, the allele frequencies are 0.875 and 0.125. Thus, random mutations can keep the allele frequencies changing. What happens if there is a continuous occurrence of forward and reverse mutations? When the rate of forward mutation equals the rate of reverse mutations, an equilibrium is obtained and allele frequencies do not change further. Equal number of individuals are converted from A1 to A2 as well as A2 to A1. So, in that case, allele frequencies do not. Another process that may bring about change in allele frequencies is the influx of genes from other populations, commonly called migration or gene flow. The overall effect of migration is twofold. First, it prevents populations from becoming genetically different from one another, and second, it increases genetic variation within populations. Here, there are two populations and the number of individuals n is equal to 24. The allele frequencies of the first population are 0.75 and 0.25. In the second population, allele frequencies are 0.625 and 0.375. Some individuals from the second population migrate to the first population. Migration may occur in search of food or to escape from predators and for many other reasons. The individuals from second population get mixed into the first one. The value of n changes for these two populations, and the allele frequencies also change. Based on the number of individuals migrated and their genotype, its effect on allele frequency may change on different conditions. Generally, migration allows mixing of genes and it keeps populations homogeneous. Different alleles arise in different populations due to rare mutational events. Because of migration, these alleles can spread into new locations or populations. Migration decreases genetic differences between populations and increases genetic variation within populations. Genetic drift is the process of random fluctuation in allele frequencies due to sampling effects in finite population. There is an equal chance for all alleles in the process of reproduction if there is no natural selection acting on it. But not all alleles get transmitted to next generation due to the sampling effect or sampling error at the time of gamete selection. Consider a population of 5 individuals with 10 different alleles. The alleles are named from A0 to A9. All the individuals are heterozygous. I have taken a random number and I will select the gametes based on the random number. So, in the first generation, even though all individuals were able to produce offspring, some of the alleles were lost. Again, I follow the random number to select the gametes. Since some of the alleles were lost, I had to omit some digits in the random number. We get the second generation of individuals. No alleles were lost. We may move on to third generation. In the third generation, we lose one more allele. Now, calculate the allele frequencies in the third generation. The allele frequencies have changed within 3 generations even though there was no natural selection acting on it. This is a chance effect. The magnitude of this drift in allele frequencies vary between populations. This means that even if natural selection do not act on populations, populations at different locations can get diverged as the time passes. Genetic drift is a major driving force of evolution. Natural selection can be identified by looking for a correlation between reproductive success and genetic variation. But if you can't find any correlation between the two for a given locus and still finds a major fluctuation in the allele frequencies, genetic drift is the reason behind. Genetic drift arises due to chance event in finite populations. But there are other mechanisms which result in genetic drift. They are founder effect and bottleneck effect. The founder effect arises due to the establishment of a population by a small number of individuals. In this picture, you can see that a few plants were the starting population in the island. The change in allele frequency in the new population was a result of the chance event. A few individuals selected by chance determines the allele frequency in the entire population. That is called founder effect. A bottleneck effect happens in a population when there is a sudden decrease in the size of the population due to some unfavorable events. The number of individuals surviving that unfavorable condition will be few and is purely chance dependent. This can drastically change the allele frequencies in the resulting population. Natural selection takes place when individuals with adaptive traits produce a greater number of offspring than that produced by others in the population. The effect of natural selection on the gene pool of a population depends on the fitness values of the genotypes in the population. Fitness is defined as the relative reproductive success of a genotype. Fitness is the reproductive success of one genotype compared with the reproductive success of other genotypes in the population. Here is a list of the mean number of offspring produced by different genotypes. To calculate the fitness for each genotype, we take the mean number of offspring produced by a genotype and divide it by the mean number of offspring produced by the most prolific genotype. There are different ways selection can operate. One is directional selection. Under directional selection, one allele is consistently favored over the other allele. As a result, selection drives the allele frequencies towards one direction, that is, towards an increasing frequency of favored allele. In the graph, you will see there were more number of light colored individuals in the starting population. When natural selection acts, more individuals are selected for the dark allele. When selection favors individuals with intermediate phenotype, it is called stabilizing selection. Balancing selection is a type of natural selection that maintains genetic diversity in a population. Two or more alleles are kept in balance in the balancing selection. Please note the difference between balancing selection and stabilizing selection. When we speak of stabilizing selection, we focus on the phenotype. Individuals with intermediate phenotypes are favored in nature. In balancing selection, more number of alleles are favored in a population. It is a concept that applies to the locus. A population with more number of alleles are selected. Frequency dependent selection is a mechanism for balancing selection. Frequency dependent selection describes the condition where the fitness of the one phenotype is dependent on its frequency relative to the other phenotypes in the population. Increase or decrease in the frequency leads to selection. In disruptive selection, individuals with extreme range of phenotypes are selected. In this graph, the plants will grow on soil contaminated with heavy metals. But such plants fails to grow on normal soil. Plants which are able to grow on normal soil are unable to grow on contaminated soil. The two extreme phenotypes were selected from the plant population.